Sara Faggio from the Department of Comparative Biomedicine and Food Science, University of Padova. My presentation will focus on the potential for integrating functional annotation data. So I will take what Luca has already presented, and I will put it in a genomic prediction perspective. So I work mainly on quantitative genetics. So my presentation will focus mainly on quantitative genetics. So just to provide a brief background. So selective breeding in aquaculture is mainly addressed to complex traits of economic importance, and common target traits are disease resistance, productivity, efficiency, and quality traits. Trait phenotyping for this kind of traits, trait phenotyping of the breeding candidates or their relatives is necessary to estimate breeding values and select the new breeders. And this is mainly what is done in traditional selection. But the fact is that routine individual phenotyping for complex traits like disease resistance and stress tolerance, feed efficiency is really difficult. It is costly, it is time consuming. So these traits are good candidates to be improved by genomic selection procedures. During the last 10 years, the technological progress have provided marker maps for different aquaculture species. And we are talking about high density marker maps. So in other words, SNP arrays covering from 1,000 to million SNPs spread in the genome of the animal. This makes individual genotyping feasible at a reasonable cost. For some species, we are also approaching whole genome sequencing, and this is very important for later. The availability of high-density marker maps allows the advancement of genetic approaches, making possible the application of genomic selection. So genomic selection means that the selection of the animals for breeding is based on breeding value calculated from the joint effects of genetic markers covering the whole genome. But but how genomic selection works? So maybe many of you already know, but let's have a quick look at how genomic selection works. So we produce a batch of fish. Then this batch of fish is randomly split into two groups of full or half sieves, the reference population and the breeding candidates. Then the breeding candidates, among the breeding candidates, we will select the new breeders through genomic selection, and we will see. So the reference population is normally tagged to track the fish throughout the period. Tissue samples are collected to perform DNA extraction and individual genotyping using high-density marker maps. We assign parentage, and we also record the phenotypes. So let's say that our trait of interest is disease resistance. On breeding candidates that are maintained safe in the farm, we perform tagging, again, tissue sampling to extract DNA, and we perform individual genotyping, and we assign parentage. What happens next? So the reference population has phenotypes for the trait of interest, disease resistance, for example, and we have also the genotypes. Phenotypes and genotypes are used in a regression model to estimate the SNP effect. Our prediction equation is that uses markers as input and predicts the breeding value for disease resistance is derived. I have already shown you that breeding candidates have only genotype. So what we do is to use this prediction equation to calculate the genomic estimated breeding value for the breeding candidates in order to select the new breeders with the best performances in terms of disease resistance. Another important part is how we estimate the accuracy of genomic prediction. That means how well perform the prediction equation we are building. So normally we try to recreate what happens in reality. So starting from the reference population, we create two groups that are the training set and the testing set. These groups are created randomly or according to the genetic relatedness between animals. So the prediction equation is built using genotypic and phenotypic information of the training set. And this equation is then applied on testing set, which we pretend we do not have the phenotypes of these animals. We just have the genotypes. In reality, we have, but we pretend we do not have. And we predicted breeding values of these animals. This is normally repeated several times. And at the end, we are able to correlate the prediction with the observed values. So let's try to summarize a little bit the advantages of genomic selection. So genomic selection overcomes problems related to the unfeasibility of breeding candidates phenotyping. So it is suitable for traits that cannot be directly measured on breeding candidates. Phenotyping needs to be performed only once for the individuals of the reference population. And the assessment of the individual genetic merit for the trait of concern becomes possible. In this way, the selection of breeding candidates can be greatly anticipated in life. And this is very useful when we are running breeding programs. But there's a but. So from our experience, complex traits of economic importance are for the majority polygenic. So how we can efficiently deal with those kind of traits? And another question is, what happens if the animals to be predicted are weakly related to the reference population? For example, they are from a different population or a different generation, a different breed. So let's start with the first question. So how to efficiently deal with polygenic traits? Polygenic traits are those traits controlled by many genes, each with a small effect. So detect these causative variants is really difficult. Theoretically, genomic selection overcomes problems related to the polygenic architecture of the traits because it estimates the effect of a large number of chromosomal regions without paying attention to the statistical significance of the effect, but trying to aggregate such effects in the prediction of the genetic merit of the breeding candidate. What we can do is improve further the prediction accuracy of polygenic traits. So we know that most of the causal variants are likely to be located in regulatory sequences. Luca has already explained that, and should impact complex traits through changes in gene expression. What we can do is determine whether the genetic variants are present in functional sequences and try to filter to create the subset of genetic markers according to that. And we can then develop prediction models that take into account biological priors. Second question, what if the animals to be predicted are weakly related to the reference population? So it is clear that testing the performance of a prediction equation that has been derived using information of a reference population that is weakly related to the individuals we want to predict, so weakly related to the target individual, is really important. What we know that has already been tested is that the prediction accuracy decreases when reference population and candidate breeders are genetically distant. So we need to use efficiently genotype to predict phenotypes. Those two questions are addressed in the FAANG projects. So the aim is to generate functional and genomic information from large population rather than using a few deeply annotated individuals. Expressed and regulatory genomic regions will be characterized. Again, Luca has already explained that. And at the end, all the obtained resources will be used to relate genotype, phenotype, and genetic merit, and predict genotype-phenotype relationships. This means that we can translate the research efforts into application, and we can promote the implementation of genomic selection in the aquaculture industry. So we can facilitate the application of genomic selection. I would like to end the presentation with a very practical example from work package 5 of the AQUA-FAANG project that is connected to the work package 3 that Luca has already presented. So the aim is to exploit genome functional annotation to improve fish breeding. In this work package, the target species is European sea bass, and the trait of interest is resistance to viral herpes necrosis. So very briefly, about 1,000 juvenile fish were produced using a commercial population, breeders from a commercial population, and fish were subjected to an experimental infection. BNN was recorded as a binary trait, so dead or alive, and as time to death. The experimental fish were genotyped using a high-density SNP panel, but their parents were wood genome sequence, so we were able to impute the offspring to wood genome genotypes. Genome-wide association analysis had been performed, and SNP markers associated to the traits were actually identified. EQTL analysis had detected loci that explains a fraction of the genetic variance of a gene expression phenotype, and active regulatory or functional elements in the genome were also detected through the open chromatin regions analysis that Luca has already shown. So, just to summarize, all the obtained resources will be used in the development of genomic prediction models, and the next step will be the integration of prior categorization of SNPs derived from functional information into genomic prediction models to predict the genomic reading values of the animals for the trait of interest, so for BNN resistance. Prediction accuracy will be determined through a validation approach, but training and testing sets will consist of animals of varying genetic relatedness according to the genomic relationship matrix. And this is connected to what I was saying before. So we would like to see how well the prediction equation perform when animals of the reference population are related to the target animals. So we would like to see if incorporating this functional information can help to the prediction equation to be more accurate, even if we are trying to predict animals of a different generation. For example, our reference population is the first generation. We would like to predict animals of the second, third, or fourth generation, or of another population. So this is really important. I think I have finished, and I would like just to thank all the people that are working on this project from the University of Padova and from the Roslin Institute. Thank you very much.